Welcome back to many of you, and thank you for joining us at EnviroLogix today for part 2 of a 3-part series on genetic editing in agriculture. My name is Jamie Welch. I'm a market segment leader at EnviroLogix, the global leader in rapid GMO and mycotoxin diagnostics. I'm pleased to introduce again our resident genetic editing expert, Dr. Emily Whiston, who will be continuing her genetic editing seminar series. Emily has worked as both an R&D molecular development scientist and has managed DNAble product development teams at EnviroLogix. In 2018, Emily has been recognized as a subject matter expert in genetic editing, presenting at the Organic and Non-GMO Forum to large global grain companies and non-GMO certification bodies. And now I'm excited to pass the presentation over to Emily.
Thanks, Jamie. I'm excited to continue talking about genetic editing, the biggest buzzword in science that has major implications throughout biology and definitely within plants and ag. In this session, we'll be taking a slightly deeper dive on the science of genetic editing, so getting into some of the nuts and bolts here of how genetic editing works in plants. As a reminder, EnviroLogix makes rapid point-of-need mycotoxin and GMO diagnostics for the global ag supply chain. Our diagnostics are sold starting with trait development and seed breeding and then through the grain handling and processing space. EnviroLogix specifically pays me to think about CRISPR because my background is in molecular genetics and comparative genomics, so that's a lot of the science that makes genetic editing possible. So I spent my PhD work looking at whole genomes, comparing millions of bases across many organisms, and thinking about the small and large changes that can impact traits on the back end. In this session, I'm going to give a brief recap on what is genetic editing that we covered in the first session, and then talk about this new generation of ag traits. So what are the molecular mechanics? Why are these traits different from other traits, and how will we be able to detect these traits? So again, as a diagnostics company, that's really our primary focus when we think about this science. So as a reminder on how gene crops were genetically modified prior to genetic editing and CRISPR, traditional breeding, crossing up plants to try to combine traits of interest, mutagenesis via chemicals or radiation to try to come up with sort of a random grab bag of new traits, some of which might be beneficial, RNAi or RNA interference, which is turning off selected genes or proteins using RNA, and transgenics, which is what has historically been thought of as GMO or genetically modified organisms, where DNA from one organism is inserted into a different organism. So continuing with the analogy of the toolbox, if each of these is a tool in a toolbox, they each do a job, one job pretty well. Sometimes you need multiple tools to get a job done around the house. But gene editing is particularly exciting for science because it combines all of those prior tools into a single multi-tool. It's very flexible, very versatile. You can do everything that those prior individual tools could do with added functionality and specificity. Genetic editing is accomplished with site-directed nucleases, and again, there are 3 major classes of site-directed nucleases. Zinc finger nucleases were the first that were published for use in biotech. Talon or talons is another. And then a lot of the sort of buzz around genetic editing has focused on the technique CRISPR or CRISPR-Cas. All of these, even though sort of mechanistically or how it gets its work done in the cell differs very slightly, the outcome of all of these is the same. There is a highly specific DNA double strand break that then goes through a natural DNA repair mechanism, and that DNA repair mechanism could give rise to certain traits of interest. How is this applied in biotech? So using the CRISPR-Cas as the example, there's a guide RNA, so that little green squiggle, which is very specific and tells the nuclease, which is Cas9, so a nuclease cuts DNA. So the guide RNA tells the nuclease amongst millions of bases which sequence specifically to make a cut. So it breaks the DNA, and then again that DNA is repaired through natural repair mechanisms that exist in the cell. DNA damage, so these types of DNA breaks, occur naturally in the cell all of the time, all organisms, all types of cells. If we think about something like UV damage from the sun to human skin, UV damage is causing DNA breaks, and then that gets repaired by our skin cells. That repair process, there are two major repair pathways. So we'll start with non-homologous end joining, where the two broken ends of DNA are essentially jammed back together in a somewhat error-prone process. And that error-prone process can create gene knockouts. So if we think about the tools, this basically replaces mutagenesis and RNA interference, but much more specifically. So the mechanism here, so end joining repair creates an indel, which is an insertion or deletion of one or more nucleotides that causes what scientists call a frameshift mutation in a gene. So how that works, so frameshift mutation creates a gene knockout. Genes code for proteins, and there are 3 bases of DNA per amino acid in a protein. So if we think about a sentence of triplets, the cat sat on a bat. So bear with me and pretend that Anna is one word. If we make a cut somewhere in the middle here, and then put the sentence back together, but we insert a new base or a new letter, it knocks out the meaning of the rest of the sentence. So it throws everything else out of frame, and now the cell can no longer create a functional protein. Knockouts don't necessarily sound like something positive, but turning off certain genes can have some beneficial impacts in plants and in animals. So here are a couple of examples that I touched on in session 1. So waxy corn is the variety of corn used to make cornstarch, and the company Corteva used CRISPR paired with non-homologous end joining to recreate a native trait, which is that waxy corn trait, in a high-yielding background. So the other example that I have here is a non-browning mushroom. So this was a non-native trait. This is what we'd call a new or a novel trait, where a scientist from an academic lab was able to disrupt the genes associated with ripening to reduce or eliminate browning when those mushrooms would be chopped and placed in a grocery store. Again, EnviroLogix is focused on detection and diagnostics. So can we detect the outcomes of non-homologous end joining? So if we take the native sequence, and we use a pair of scissors, and we make a cut, that cut is repaired. And so now in this sequence, we have those 2 little gray plus signs. There has been an insertion in that native sequence, and that is detectable. So molecular methods, PCR or EnviroLogix rapid molecular technique DNAble can specifically amplify and detect very small differences in DNA sequence. There's a little caveat on there. So we can detect it if we know the specific sequence that was targeted and changed. Because again, DNA damage and resulting minor changes occur naturally all the time. So unless we or somebody developing a PCR assay knows exactly what might have been the outcome of biotechnology or bioengineering versus the natural process, it would be very difficult to pinpoint individual changes that were the outcome of genetic editing versus something that was just it sort of naturally happened to arise in the cell. So thinking about the other repair pathway that can put broken DNA back together, that would be called homology-directed repair, where a piece of donor DNA is provided by a scientist, and that tells the cell very specifically how to repair the broken ends of DNA. And this can be used for sequence insertion or editing. So again, if we think about the tools in a toolbox, this basically can be used to do almost anything in a cell. It's a challenging technique, but it's extraordinarily powerful. So this could be used to replace some traditional breeding. It can be used for RNAi. It can be used for transgenics. Any combination of the above and more beyond. So edits could be multiple bases or down to a single base change, again, out of millions of bases in a genome. Or this can be used for insertion of new sequence, whether that's a promoter that influences gene expression, a whole new gene, or RNAi. If we start with editing, what can a SNP edit do? So a SNP is a single nucleotide polymorphism. So a single base pair change. So here's an example, and this is highly theoretical because this is actually a paper that was talking about the SNP itself and was not about editing. So these scientists sequenced rice and discovered that there's a native SNP, so a single base pair change, that confers cold tolerance in rice. So if someone wanted to recreate this cold tolerance in a new variety of rice, in ag we'd call that precision breeding. So the rapid introduction of desired native traits without yield drag. And CRISPR paired with homology-directed repair could very specifically recreate this native single base pair change in a different type of rice. Are these types of targeted SNPs detectable? So if we start with the native sequence, make a cut, and then make a change so that what was green, now we have a single base pair change to blue. Is that detectable? Yes. So molecular methods, again, PCR and DENABLE, can very specifically amplify and detect extraordinarily minor differences in DNA sequence. But again, there's a star. There's still a caveat here. If that is a native allele that occurs naturally in the population of the same plant, it's extraordinarily challenging, potentially not possible, to say which is the native allele versus which was the outcome of CRISPR paired with homology-directed repair. So again, if that single SNP was a native trait that was moved between varieties of the same plant, that becomes nearly undetectable. So sequence insertion, the other possibility from homology-directed repair. So that's taking the native sequence, making the cut, and the repair introduces a much larger block of new sequence. So that's that whole blue chunk. So that could be that blue new DNA, could be transgenic, so DNA from another species, or it could be cisgenic, DNA from the same species. Again, this could be a promoter that changes gene expression, or it could be a whole new gene that adds or changes functionality. So what's the point of cisgenic insertions? I mentioned this example of cows in session 1. So this is where scientists took an ancient allele or a native piece of sequence from beef cows, and that sequence conferred the polled trait, so that's a naturally hornless trait. And they took that sequence and inserted it into elite line, high milk producing dairy cows, so that those dairy cows, the offspring would be born with the polled trait. They would not be born with horns, and they would not have to go through the dehorning process, which is pretty costly, pretty painful to the animals. So it would definitely be desirable to have these high milk producing dairy cows that don't have to be dehorned. Again, this would be an example of precision breeding, so that rapid introduction of desired native traits without yield drag. Is this detectable? So I hope from the last example that, the automatic answer is yes. So again, the molecular detection techniques, PCR, DENABLE, can detect the junction between native and inserted sequence. So there's a difference between green and blue here, and that is detectable by molecular methods. And then protein detection methods, so lateral flow strips, which makes up the bulk of the EnviroLogix product portfolio, can detect transgenic proteins. So again, proteins or genes from a different species that have been inserted into a plant. Still a caveat here, because If we think back to that example with cows, even though it wasn't a single base pair change, there was a whole insertion of native sequence from one type of cow into a different type of cow. This could certainly be accomplished in plants as well. So a native allele that occurs naturally, again, it's nearly impossible to tell the difference between a native trait and an engineered trait if it's simply recreating the exact same sequence in a new variety. So some key takeaways for this slightly deeper dive on the science of genetic editing. So editing mechanics can include gene knockouts and versatile genetic insertion or replacement. Why is this different from other methods? So it's significantly faster, it's more flexible, and more precise than traditional methods of genetic modification. And how will we detect this moving forward? So most edited traits can be detected by a molecular DNA-based method, so PCR or DNAble, but this definitely presents new challenges for the diagnostics industry, and we hope you'll tune in to part 3 to hear more about that. So with that, I'll say farewell from Maine, where sometimes it's cold. It's certainly very cold right now, but it's always beautiful. So if you ever want to come visit, let us know. And if you have any questions, please do feel free to email me. Thanks so much for listening.